MIB Agents OsteoBites webinar and podcast presents the latest in osteosarcoma treatment, research, innovation, and hope each week. Today, we are talking with Dr. Robbie Majzner on unleashing the pediatric immune system against osteosarcoma and other solid tumors. On the panel are physician's assistant Amy Woodchecky, a childhood cancer survivor, and 2021 MIB junior board members and osteo warriors Charlotte Murdoff and Mia Sandino. I'm your host, Anne Graham. Welcome to OsteoBites, everybody. Get your snack, get your coffee, let's go. It's a great day to talk to Dr. Robbie Majzner on unleashing the pediatric immune system against osteosarcoma and other solid tumors. Dr. Majzner is an assistant professor of pediatrics at Stanford University School of Medicine. After graduating with a BA from Columbia University, he attended Harvard Medical School, where he developed an interest in pediatric oncology. Yay, thank you for doing that. He completed his residency at NY Presbyterian-Columbia and his fellowship in pediatric hematology-oncology at Johns Hopkins and the National Cancer Institute. During his fellowship, he cared for some of the first pediatric patients to receive CD19 chimeric antigen receptor, or CAR T cells. Witnessing the success of CAR T cells in these patients drove Dr. Majzner to the laboratory, where he focuses on developing immunotherapies for children with incurable solid tumor cancers. His laboratory work aims to generate highly efficacious CAR T cells and to find new immunotherapeutic strategies to treat pediatric cancers. So happy you're doing that. Our panel today includes 2021 junior board members, Charlotte Murdoff and Mia Sandino. And we also have Amy Woodchecky, a childhood cancer survivor and pediatric oncology physician's assistant. I'm your host, Anne Graham, president of MIB Agents. MIB Agents is a pediatric osteosarcoma nonprofit dedicated to making it better for our community of patients, caregivers, doctors, researchers, with the goal of less toxic, more effective treatments and a cure for this aggressive bone cancer. MIB makes it better for kids in 3 ways: through direct patient and family support, with many programs to ensure that no one walks alone through this disease; through education, including our annual Factor Conference, OsteoBites, our testing and research directory, and our book, Osteosarcoma, From Our Families to Yours. If you or your institution would like some of these books to hand out to patients, or you need one for yourself, let us know. It's in Chinese, Spanish, and English. And finally, through research, by funding it, sharing it, and supporting our researchers and physicians. Tomorrow, by the way, speaking of programs, is the last day to sign up your Osteo Warrior for Warrior Mail. Agents across the globe handwrite letters of support and cheer for those in treatment, and there might also be some swag included. Go to our website for all the intel, just look up Warrior Mail. With that, Dr. Majzner, would you get us started by introducing yourself, please?
Hi, everyone. I'm Robbie Majzner. I'm a pediatric oncologist and physician scientist at Stanford. And I just am very excited to be here. And Anne, just want to thank you. And, it's so amazing to see such a great advocacy group coming together and growing around osteosarcoma.
So, hi, my name is Charlotte Murdoff. I'm a 3-time osteosarcoma fighter, currently undergoing chemotherapy treatment for my 4th relapse, and I just finished my 5th cycle out of 6. And I was originally diagnosed in July 2018.
All right. Hi, my name is Mia Sandino. I am a sophomore at the University of Washington. Go Dogs. I have been battling osteosarcoma since September of 2018. And I am currently pursuing my BS in psychology at the University of Washington with hopes to become a clinical psychologist to work with AYA cancer patients, much like myself and Charlotte. So.
Hi, I'm Amy Woodcheck. I'm a physician assistant in pediatrics for about 16 years now, and the last 10 or so have been in pediatric oncology. I'm also a survivor of leukemia 36 and a half years ago, and my passion is just to make it better for these osteo kids because we got to beat this somehow.
So Yeah, I'll speak today about, starting to use immunotherapy for osteosarcoma. And, I talk specifically about the pediatric immune system because I think in pediatric diseases and in children, the immune system, the way it interacts with cancer is really fundamentally different, we're learning, than with adults. And this has emerged really as an important, dichotomy in the last 10 years as kind of immunotherapy has become the soup du jour of oncologists, especially in adults, where there's been a lot of success in some adult solid tumors. So I'll talk about immunotherapy for pediatrics versus adults a little bit. I'll give you then two research kind of vignettes, one around B7H3 targeted CAR T cells, which we think is a great target in osteosarcoma. And then the second around reorienting the immune system and specifically macrophages against osteosarcoma and neuroblastoma. So, the unfortunate pediatric oncology in a way has been a huge success story, especially when you look at really leukemia, where over the last 40 years, we've learned to cure the majority of patients that are diagnosed with ALL. And when you look at solid tumors, clearly the progress has not been as great, or really, when you look at these Kaplan-Meier curves, you see that there's basically been no progress in patients that have been diagnosed with metastatic solid tumors. So despite over the last 40 years kind of intensifying our therapy between more chemotherapy, radiation, more advanced surgery, and other therapeutics, we've really seen the needle not really move for some of these sarcomas and solid tumors. And an exception to that has been the solid tumor that occurs almost exclusively in pediatrics, known as neuroblastoma. Where you've seen this steady uptick of survival of patients that are diagnosed with high-risk metastatic neuroblastoma of about 10 to 15 percentage points every year, to the extent that the last COG study now had an event-free survival of nearly 70%. And there's been a lot of things that have made a difference here, but one of the biggest things is the integration of an anti-GD2 monoclonal antibody, so an immunotherapy that was integrated into upfront therapy for neuroblastoma. And this really kind of was the first time, it was actually the first drug ever specifically approved for pediatric cancer, and was the first time that immunotherapy had made it into the upfront therapy for a pediatric solid tumor. So, what we're hoping to do in the lab is kind of apply this new treatment paradigm to cure more metastatic solid tumors. And when we think about cancer immunotherapy, we really like to divide it into kind of two different worlds. One of them we call immune response amplifiers. And this world of immune response amplifiers has really exploded in adult malignancies. So there's this class of molecules called checkpoint inhibitors, which I'll talk about. And these basically unleash the body's the immune system's natural T-cell based immune response against cancer. And in adult tumors, you've seen use of these inhibitors. So there's PD-1 inhibitor, CTLA-4 inhibitor, and using these checkpoints, we've seen a real change in the way that diseases like melanoma and lung cancer are treated. And for the first time, we're seeing cures of adults that have stage 4 metastatic melanoma with this kind of immunotherapy-based approach. Now, in order to unleash the immune system, the native T cells against a tumor, they need to respond to something. And T cells basically are programmed to respond to genetic mutations that happen on the tumor, foreign proteins or mutated proteins that occur in the tumor. And I don't show it here, but in adult malignancies such as melanoma, you have very, very high rates of mutation. So above 100 mutations per megabase. So this is a high mutational rate in things like melanoma, lung cancer, which are driven by environmental exposures such as cigarette smoke, sun, etc. And as the tumor builds up all of these mutations, it basically starts presenting new antigens, neoantigens, to the immune system. And by giving these checkpoint inhibitors that have really changed the day for adult malignancies, those unleash the T cells to respond better against those mutations. Now, this is data from a group in Toronto, Adam Schline's lab, where they looked at the rate of mutation in pediatric cancers. And unlike adult cancers, pediatric cancers, you can see that only 1% are really have this really high level of mutation. And only an additional 4% have kind of a moderate level of mutation. And the vast majority of pediatric tumors have a very low level of mutation. And this makes sense with what we know biologically about pediatric tumors, which are really oftentimes driven by fusion, single gene fusions like EWS, FLY1, and Ewing sarcoma, or epigenetic changes that occur. And we don't have high rates of mutation in pediatric tumors. So we also looked at PD-L1 expression across a lot of kind of pediatric sarcomas and some brain tumors, and PD-L1 expression is oftentimes seen as a biomarker for response to anti-PD-1 therapy. And as you can see in these pediatric tumors, almost none of them expressed PD-L1. And there was a recent trial of anti-PD-1, one of these checkpoint inhibitors, nivolumab, through the Children's Oncology Group in several pediatric solid tumors. And you can see here in Ewing's, rhabdo, osteosarcoma, and also neuroblastoma, there really were, unfortunately, no patients who had an objective response to the single agent nivolumab. So this is really most likely because pediatric tumors are fundamentally different. They don't have high rates of mutations, and they're unlikely to respond to single agent checkpoint as kind of a single maneuver. So because of that, we've really focused on the other end of the balance here, which are synthetic immunotherapies. And these are things like monoclonal antibodies, or what I'll talk about today are CAR T cells, so things we can make in the lab that can initiate a new immune response. So it doesn't just turn on the natural immune response, but it basically starts a whole new immune response. So CAR T cells basically are a hybrid between an antibody, which can bind to a target or surface protein or surface antigen on a tumor cell in a very specific manner. They'll only recognize that one target. And it's combined with the signaling domains from the T cell receptor in order to have this, this very specific cytolytic T cell response against tumor cells that express the target of that antibody. And this T cell receptor basically is restricted to look for things that are mutations within the cell as expressed by the tumor cell in MHC. And as I mentioned, pediatric tumors really don't have those mutations in MHC. And so the T cell receptor generally has been ineffective in kind of in trying to push this T cell receptor to respond to pediatric tumors. However, CARs can recognize surface antigens. So these are antigens, not things that are mutated in the cell, but might just be overexpressed on the surface of a tumor cell. And obviously this technology has been extremely successful in pediatric leukemia with CD19 CAR T cells. So here is Emily Whitehead, who was the first pediatric patient treated with CD19 CAR, had a complete response and has supposedly active CAR T cells in her blood more than 8 years from the first time she was treated. And this is data that is the results from the seminal trial of CD19 CAR in pediatric ALL run by Shannon Maud and the folks at University of Pennsylvania and obviously at Novartis and through a multi-center trial, which really showed amazingly that basically 50% of patients that were able to get this therapy ended up having long-term event-free survival. So we put them into remission and they would stay there. So really fantastic results for CD19 CAR. And this is about the time that I was doing my my fellowship when these trials were first being run. As Anne mentioned, that we were running another CD19 CAR trial at NCI that I had the honor to take care of the patients on that trial. And I saw that technology and I said, oh, this is just amazing. You really can hijack or hack the T cell to recognize a tumor cell and just send it on its way. And we said, okay, let's just try to, I joined Dr. Crystal Makel's lab with the intention to say, okay, let's figure out how we get these to work in solid tumors. So, it's been a long road and lots of failures and successes, but through the failures, we learned a lot, I think, and are really learning how to make these receptors work better. But I'll just tell the story about one specific receptor that we generated that we think has promise in solid tumors and specifically in osteosarcoma. So there's a target known as B7H3, and this is a checkpoint molecule that's expressed on many tumor cells and has been really well known to the pediatric oncology community because the group at Sloan Kettering has been targeting it with a radiotherapy conjugated antibody for more than 10 years. So it's been a well-known target that's been expressed, all over pediatric solid tumors and other solid tumors. So we said, okay, this could be a good target for CAR T-cell therapy. And so what we did, we screened tissue arrays of all different pediatric tumors. And you can see that we found that we had very frequent expression of this target B7H3 on rhabdomyosarcoma, Ewing sarcoma. We don't show osteo here, but it's also frequently on osteosarcoma. So ultimately, we found that more than 80% of pediatric tumors express B7H3. And then importantly, when we went and looked at how, at the level of expression, we saw that it's often very high and homogeneously expressed, at least on our diagnostic tumor samples that we have here. So you can see this kind of very intense brown staining, indicating that there's a lot of the protein present. So, armed with that data, we said, okay, let's make a CAR against B7H3. So as mentioned, we have the signaling domains in the T cell, but we need an antibody that recognizes B7H3. So fortunately, this company known as Macrogenics that's works out of Rockville, Maryland, they had developed a B7H3 antibody and specifically found that it stained, these are adult adenocarcinomas, prostate, breast, etc. It stained quite intensely on those tumor types. And then when they went and looked at normal tissue, even some normal tissue that's known to express B7H3, like the liver, they found very little staining. So it had a very preferential staining to tumor-associated B7H3. And in working with that company at the NCI, we made a CAR using this binder known as MGA271, using some of the signaling domains that actually make up the Kymriah CAR that has been FDA approved for B-cell ALL. So, we actually ended up screening 10 different antibodies for B7H3 and a bunch of different formats of the CAR, and this one ended up kind of being the winner. So when we went into kind of animal studies, so here we use the MG63.3 cell line, which is an osteosarcoma cell line that's highly metastatic also. We see that the CAR T cells can make cytokine, which is what they're supposed to do when they see their tumor cells. But more importantly, we injected the MG63.3 cells in the hind leg of the mouse and basically let it grow for 3 weeks until you have a real, palpable tumor, a measurable tumor. And then we intravenously treat the mouse with a single dose of B7H3 targeted CAR T cells through the tail vein. And as you can see here from these are curves of the tumor growth, you can see that when we treat with a B7H3 CAR, you basically get regression in all mice that receive the B7H3 CAR. The tumor comes down and it doesn't reappear. And mice that get an irrelevant control CD19 CAR, there's no change in tumor growth, and the mice ultimately die of their large tumors on their hind leg. So this tumor cell line also is very metastatic. It was specifically selected because it has a high metastatic potential. So in this model, it's a little bit gruesome. I apologize. We inject the tumor cells into the hind leg, and then we wait a month. And so the mouse at that point has a pretty large tumor, and then we amputate the hind leg with the tumor. At that point, all the mice have micrometastatic disease. They have small bits of osteosarcoma spread throughout their lungs. We then let them recover from surgery and then treated them with CAR T cells. And what you see here is that mice that received the B7H3 CAR T cells, we were able to completely prevent their death from metastasis over the next 6 months, whereas mice that didn't receive any treatment, they died over the next 60 days. And this is just when we've taken out some of the lungs from the mouse who went untreated, you can see that they just have these huge metastaticness of cells that had really started in the leg and been able to metastasize individually, to individual nests in the lungs and then grow up over time. B7H3, as I mentioned, is really expressed broadly both on solid tumors and brain tumors. So here I just show that we had a similar effect in Ewing sarcoma, and then we also saw the same effect in medulloblastoma in this original paper. And then there's been a follow-up paper also looking specifically in another type of pediatric brain tumor that occurs in infants known as ATRT, that they're quite effective also. So, we're excited about the potential of this therapy, but of course, as I mentioned, B7H3 is also expressed on normal tissues at very low levels. And so we wanted to kind of model, okay, does our CAR recognize those low levels? Do we think we have a therapeutic window? Meaning, do we think that we can have generated CARs that are going to attack normal tissue that expresses low levels of B7H3? Or might we get lucky and our CAR T cells really can only see the high levels of B7H3? So here we generated some cell lines that express low, medium, or high amounts of B7H3 on their surface. And then we just, this is a tumor cell killing assay where you can look at the rate of killing of those cells by B7H3 CAR T cells over time. And you can see that the CAR T cells really only kill the very high B7H3 expressing tumor lines. The medium lines, they kind of control, and the low lines, they generally leave alone, and looks basically like the negative, the negative control line. And then when we went into mice and we went against, we tested this B7H3 CAR against our leukemia cell lines on which we had artificially expressed high B7H3, we see that the CAR T cells are quite effective. But when we express low levels of B7H3, the CAR T cells basically have no effect. So, this is obviously not a perfect model for toxicity in a human, and we'll really only find out in phase 1 clinical trials whether this is safe. But we think that hopefully something about this CAR kind of opens a therapeutic window for B7H3, such that low B7H3 expression on normal tissue may be tolerated, and the CAR T cells may not recognize that low level, whereas they can go after tumor that has high level. Now, of course, this opens really the Achilles heel of immunotherapy, in that tumor cells are just really, really smart. We've seen it with CD19 CAR in leukemia, that leukemia comes back no longer expressing CD19. So if the T cells are no longer able to express, if the T cells cannot recognize low levels of B7H3, you can anticipate that eventually tumor cells may figure out kind of how to mutate around this. And it'll be a race for us to figure out exactly how to do this, how many different things we need to target at the same time in order to be effective. So that's kind of my first story. So why don't I stop there and answer questions. I'll just summarize to say we think B7H3 is a promising target. We have a CAR that has some potent anti-tumor activity, and we hope it may spare normal tissues that express low levels of B7H3, both because the antibody seems to preferentially bind tumor, and that's the data from the company, but also because something about the CAR structure limits its activity when B7H3 is low. We're currently planning a clinical trial at Stanford. We'll begin with local delivery into brain tumors, but fortunately, Seattle Children's has an ongoing B7H3 CAR T cell trial. Actually, they have two, one for brain tumors locally delivered into the CNS, but one of them is currently in dose escalation given peripherally for patients with solid tumors, including osteosarcoma. So people that are interested in learning more about that trial, you can check clinicaltrials.gov and read more about the trial and contact the PI. But we're excited that this approach has already reached the clinic.
I have a question for Dr. Majzner. I was just wondering, I know that it takes a significant amount of time to get the CAR cells ready. If you're talking removal surgically and then CAR cells, what's the time period, that time, and how could that be applicable to like human models? Would you then proceed with chemotherapy, or is the goal to eliminate the chemotherapy?
Yeah, that's a great question, Amy. I think we all dream of the day we recognize that chemotherapy is kind of a hammer for something that needs something much more precision. And, I'll hope for the day that we're not using chemotherapy in any patients. I think right now, the way all of this is being developed is for patients because they're phase 1 data, they're phase 1 trials, and we need to learn how safe and efficacious they are. It's being tested in the relapse setting. And I think you get at a great point. A lot of time a patient relapses and they need a therapy right away. And a huge downside to CAR T cells is that it takes somewhere between 2 weeks and 3 months to make the actual cells. And it depends on the process and the company that does it, or the company or the institution that does it, and the waiting. There oftentimes are just long waiting lists, and there's not enough infrastructure to make the amount of CAR T cells that people want to make right now because it's such a new technology. Some of that will get better as new technology comes out. There's companies that are saying they can make CAR T cells in 4 days now. There's also off-the-shelf versions of CAR T cells, and as those technologies get developed, we think it'll help do more trials, accrue the trials quicker, and most importantly, help more patients. For now, I think mostly this type of trial would be for a patient who's relapsed, and probably, it depends on the specific situation of the patient. Some patients, they relapse and they can wait a month or two while therapy is made, and other patients, they'll need to kind of get their T cells taken out, and while they're being made, receive some chemotherapy.
A parent is asking, their child with lung metastasis was negative for PD-L1 and TILs, and is currently on Votrient or pazopanib. Is there other options or therapies to look for in immunotherapy right now, and where should they look for that?
So that's a great question. I think that all the available immunotherapies for osteosarcoma now are really experimental and need to be done in the context of a clinical trial. And I think that your doctor can help you find some of those trials. I'm also always happy to help if anyone wants to reach out with suggestions about possible trials that I know about. So,
for B7H3 CAR T, the preclinical data is just really exciting. So I know that you said that at Seattle Children's, they're doing a clinical trial right now, and that at Stanford, you guys are doing one in brain tumors. But are there any other upcoming trials at other institutions that you know about so far?
Yeah, so specifically for B7H3, there's also a CAR T cell trial that'll open in at St. Jude at some point in the next year for solid tumors. And I'm not familiar with all the specifics about which diseases specifically they're going to treat. And I assume it will be all solid tumors, but I'm not sure about that, and when exactly they're going to be open. But we could reach out to them also to find out more. There are some other groups, one at UNC who have developed B7H3 CAR T cells, and I'm not sure exactly what their plans are, but I think over the next year or two, we will see more and more trials open. There also are other CAR T cell trials open. There's GD2 CAR T cell trials open in Texas. At Texas Children's. For osteosarcoma, we'll be opening the GD2 CAR T cell trial at Stanford and 8 or 9 other centers, a multi-center trial. But hopefully, by midway through this year, maybe a little bit later, depending on some issues that are still being worked out around manufacturing. But there are other trials open in other places, and I really, urge people to speak to their physicians about it, and in order to kind of identify trials that are good for them.
Aside from resistance, is durability an issue? And how long do CAR T cells stay in the system, and does it require multiple infusions?
So it's a great question. Thus far in pediatric leukemia, it seems about 30% of total patients can get a single infusion of CD19 CAR, and those CAR T cells last anywhere, can last in those patients for 6 months or many, many years. And it seems that some level of durability is required in pediatric leukemia. And we assume for solid tumors, it'll be the same, and durability will be an issue. And I think, it is definitely another potential resistance mechanism, is that CAR T cells could kind of go away over time, disappear over time, and you could potentially require multiple treatments. There was a really interesting case report from Baylor, from Nabila Med's group, where they treated a patient with HER2 CAR T cells that had metastatic rhabdomyosarcoma, who went into remission, I think, after the 6th infusion, actually, and then the disease eventually returned, and then had another infusion that induced a remission. So I think, honestly, we don't know the answer yet, and we're still figuring it out in these trials.
Is immunotherapy only appropriate for metastatic cancers? And is it always a follow-up to first-line chemotherapy?
Right now, when an immunotherapy like a CAR T cell is experimental, yes, it's really only appropriate for a relapse cancer, because if a patient is curable with kind of known standard of care, there's no indication yet that CAR T cells could be could be more effective and less toxic than standard of care. That said, that's clearly the goal. In leukemia, the Children's Oncology Group already has a trial open for patients that don't respond to their first 2 or 3 cycles of chemotherapy will get Kymriah, the Novartis CD19 CAR. So I think there's a lot of, once we find safe and efficacious therapies, obviously, as researchers, we're all aware of the huge burden of late effects that is associated with chemotherapy, especially in osteosarcoma, and really want to push these immunotherapies up front and sooner. Back to kind of this two-pronged approach to cancer immunotherapy. And I said that because checkpoint inhibitors in pediatrics really haven't been the home run that they've been in adult malignancies, we've really focused on synthetic immunotherapies like CAR T cells. But I'm going to go back to the immune response amplifiers for a minute, for a few minutes, and just talk about a different cell type other than T cells that really infiltrate pediatric tumors. So this is a paper from about 15 years ago where all they did was take a bunch of pediatric tumors and adult tumors and compare the immune infiltrate. And, the first thing they found is that there's more T cells in adult tumors. And this is not surprising based on what we had talked about low rates of mutation in pediatric tumors. But the next thing was that CD68, this is a marker for myeloid cells and macrophages. And macrophages are a type of immune cell that basically eat tissues and eat cells that are supposed to be removed. And we've always known that there are these high levels of macrophages in pediatric tumors and never really understood exactly why they're there. Part of it is people say they're immunosuppressive. They're suppressing things like T cells and NK cells, and they're kind of deployed by the tumor in order to evade the immune system. Or they may just be there because there's a high rate of turnover in these rapidly dividing tumors, and they're just basically eating dying tissue. And we've never really known. So there's a story that was developed by Irv Weissman and Robbie Majzner at Stanford, where they identified this molecule that's expressed very highly on tumor cells and expressed on a lot of normal tissue also, known as CD47. And they discovered the role of CD47 is to interact with this molecule SIRP-alpha, which is it's basically a checkpoint molecule for macrophages. And like blocking PD-1 would tell T cells to activate, if you block this axis, the SIRP-alpha CD47 axis with an anti-CD47 antibody, you basically block this don't eat me signal, and you tell the macrophage simply to eat the tumor cell. So the decision for a macrophage to kind of eat the tumor cell or not is really a balance of several factors. There's this one I just talked about, CD47 and SIRP-alpha, but then there's some prophagocytic or eat me signals. And those include something as simple as an antibody that can bind to the cancer cell and provide this Fc portion of the antibody, which interacts with a special receptor for antibodies on macrophages known as the Fc receptor, and that drives phagocytosis. But then there's this other molecule, calreticulin. This is a prophagocytic or eat me signal, and it's basically expressed by dying cells when they're ready to be removed by the immune system. So calreticulin is usually expressed inside the cell, and it's shuttled to the surface of a cell when it's ready to be removed. And this happens on cancer, but it also happens on cardiac heart cells after a heart attack also, and any cell that really is going to be removed by macrophages. So when the macrophage is making its decision to enact anti-tumor activity, to either eat or don't eat the cancer cell, it's really a balance of all these different receptors. And I'm going to talk now a little bit about one way we figured out how to kind of switch that balance. So a few years ago, the company that had been spun out of Stanford that made the first anti-CD47 antibody presented this data where they took their anti-CD47 antibody and they combined it with an anti-CD20 antibody. And this antibody, rituximab, is a very commonly used antibody in patients that have lymphoma. And they gave this combination to patients who had lymphoma, but who were already resistant to rituximab. So they gave it in combination, basically reasoning that, okay, maybe they're resistant to rituximab because of high levels of CD47. And if we give anti-CD47 and rituximab, we'll see patients have a response. And, it's pretty amazing. They reported nearly a 30% complete response rate across 2 different types of lymphoma. And, that's pretty good for just kind of 2 off-the-shelf antibodies. And so when I saw that data presented at ASCO, I said, okay, what's really the only antibody we use in pediatric oncology other than rituximab? And it's this anti-GD2 antibody that we use in high-risk neuroblastoma. And GD2 is a target that's really well known in pediatric oncology because we have this drug that's FDA approved to treat neuroblastoma. But GD2 is also expressed on a wide variety of other pediatric malignancies, including osteosarcoma, and I'll talk about that in a minute. So this was one of those projects that we started with the in vivo project, in vivo experiment in the mice, and then we saw really synergistic activity between anti-CD47, which basically had no effect to control these tumors, these neuroblastoma tumors in the mice, and anti-GD2, which also had almost no effect if the tumor was large, and had a small effect if the tumor was kind of small or tiny. But when you combined GD2 and CD47, you basically ended up curing all the mice. And we ended up doing this across 3 different models, and then even in an immune competent syngeneic model. And you see here that only the mice that got the combination of anti-GD2 CD47 have kind of durable disease control. And even the mice that had initially responded to anti-GD2, those tumors eventually grow out. And so you really just have this beautifully synergistic combination of the synergistic activity between these 2 antibodies, anti-GD2 and anti-CD47. So we wanted to understand more about what was driving that synergy. So we went back to kind of in vitro assays. And so here, this is just an assay that shows the amount of phagocytosis when you take human macrophages and tumor cells. And you basically can look up into this corner on each of these graphs to see how many macrophages are actually eating tumor cells. And what you see is at baseline without antibody, you have almost none. You add anti-GD2, you get a little bit. You add anti-CD47, you get a little bit. But you get this nice synergistic activity that's more than just adding the 2 up when you add the 2 antibodies together. And that's really what we wanted to understand. Why did GD2 and CD47 go together so well? So we said, okay, maybe it would happen with any antibody. So we got another antibody that recognizes B7H3, and we combined it with anti-CD47, and we saw no synergy between B7H3 and CD47, either in that phagocytosis assay or also in mice. As you can see here, the anti-B7H3 with CD47 is no better than the anti-CD47 alone. So we said, okay, there must be something that's really GD2 specific. So, we went into the literature reading about GD2, and it's an interesting phenomenon that's been described in a handful of papers, that when tumor cells that have high GD2 are treated with an anti-GD2 antibody, even if there's no immune cells in the dish, the cells start dying. So you see here a couple cell lines, and they just kind of show how the cells kind of start bunching up and eventually die because there's some kind of cross-linking of GD2. There's some kind of signaling that happens as a response to this antibody. And now that's pretty rare. Most antibodies are thought really just to either kind of tag the cell for removal by the immune system or to block kind of important signaling mechanisms. But this is driving some kind of signaling inside the cell that's driving cell death of the tumor cells. And so we went and we looked, and these are a couple neuroblastoma cell lines, but we saw the same with osteosarcoma. Also, when we treated with anti-GD2, we got some amount of cell death. But then we looked at the cells that survived, so those cells that didn't die, and we looked at their expression of that eat me signal, that prophagocytic signal, calreticulin. And what you can see here in blue for tumor cells that were treated with anti-GD2, they have this huge increase in the amount of calreticulin on their surface. So they're basically screaming to the macrophages, screaming to the immune system, eat me or remove me. And we saw that across tumor cell lines, and it didn't happen when GD2 was knocked off the surface. So we knew that this was GD2 specific. So just by adding anti-GD2 antibody into the mix, you're basically driving kind of stress signaling in tumor cells that signal to the immune system, okay, it's time to act against me and to remove me. And then we wanted to look from the other end. Was there something that GD2 was interacting with on immune cells? So with a collaborative lab in Carolyn Bertozzi's lab with Benji Smith, who's a MD PhD student, we screened to look at these molecules known as Siglecs. So these Siglecs are basically they're receptors that are expressed across human immune cells. And they're most commonly inhibitory. They have something that is called an ITIM domain, and they're expressed, some of them on macrophages, some of them on T cells, some of them on B cells. And we said, okay, Siglex basically by definition binds sialic acid, and GD2 is what's called a disialoganglioside, which is basically a fancy way of saying it's a sugar that contains two molecules of sialic acid. So we said, okay, if something on immune cells is binding GD2, it's got to be one of these Siglex. So we needed to make a cell that didn't express any sialic acid other than GD2. So what we did was we took a cell line that's GD2 negative, and you can just treat it with chemicals called sialidase that basically just rips all of the sialic acid off the surface. So now you have a tumor cell that is basically naked as far as sialic acid. Then you can just take some GD2, which a company somehow gets out of some cow brains, and you just put it in a dish with these cells, and all of a sudden you have a tumor cell line that's covered in only GD2. It's the only sialic acid containing molecule that's expressed on this tumor cell. And then what Benji did was take each of the Siglex, so you can buy kind of recombinant forms of each of these Siglex. So instead of them being expressed on immune cells, you just have ones that you can probe with, and you can basically conjugate a fluorophore so that you can detect it afterwards by flow cytometry. And what he found was amazing is very, very specifically, this single Siglex, Siglex 7, binds the synthetic GD2 that we put onto the that we had loaded onto our tumor cells. And now Siglex 7 is one of these inhibitory Siglex. It works against immune cell function, and it's exclusively expressed on NK cells and macrophages. So this we knew we had found something important because those two cell types are basically the only ones that really infiltrate most neuroblastomas. So then we went and looked if Siglex 7 binding to GD2 could be blocked by an anti-GD2 antibody. And that's exactly what we found, both on our tumor cells that artificially express GD2 or on these neuroblastoma cell lines, that giving the anti-GD2 antibody basically block the interaction between Siglex 7 and GD2. So what does this mean? It means that basically GD2, like CD47, is also one of these don't eat me signals, that Siglex 7 is expressed on macrophages and on NK cells and inhibits their function, and when it's ligated by GD2. And by using an anti-GD2 antibody, we're also driving kind of this this blocking of a don't eat me signal and driving phagocytosis. So when you think about the balance between don't eat and eat that we have in our system here, now what we've done is we basically use two antibodies in order to block two different don't eat me signals that's coming from the cancer cell. And we're pushing towards phagocytosis. We also, the anti-GD2 antibody is providing an FC to the FC receptor on the macrophage that's a pro-phagocytic signal. And then that stress signaling that's happening because of the anti-GD2 antibody it's happening inside the tumor cell is shuttling calreticulin to its surface, which interacts with this the LRP receptor on the macrophage. And in the end, you just squarely push towards phagocytosis and really unleash the macrophages. So why am I talking about a neuroblastoma project on this osteosarcoma call? It's because GD2 has been known for a long time to be expressed on sarcomas, but very commonly on osteosarcoma and less so on things like Ewing sarcoma and rhabdomyosarcoma. So we've always been interested in using anti-GD2 to target osteosarcoma and find a way to do it. People have used the antibody in multiple trials, the antibody alone, like it's used in neuroblastoma, and there have not been clear signs of efficacy yet from the anti-GD2 antibody. So, we think we were hopeful that maybe we could add CD47 and see some effect. So that's exactly what we did. Here we did the model where we injected the cell line in the hind leg, and then we let it grow for a week, and then we treated with either GD2, 47, or the combination. And you can see in purple, only the mice that got the combination of GD2 and 47 had any disease control, and the tumors didn't grow out for a long time, for about 2 months, at which point they started growing out, but they actually had all lost GD2. So, heterogeneity, again, will end up playing an issue here. But what this showed us was that GD2 alone and CD47 alone are really ineffective in this disease. And it's only when you have that combination that really drives or reorients the balance towards phagocytosis do you start seeing anti-tumor activity. And then, what we're even more excited about is what happened in that metastatic model, where again, we let the tumor grow for a month. Then we cut off the primary tumor with the hind leg. And at that point, the mouse has lots of micrometastatic, very small metastases in their lungs. We think this really represents, the actual condition that happens in children and young adults that have osteosarcoma, many of whom have those mets at the time of diagnosis. And that's why even chemotherapy is effective. It's mostly clearing the metastases that have already taken kind of hold in the lungs. So then we treat with those antibodies, GD2, 47, or the combo. And then after a month, we take the lungs and we look at them. And what we see here is that mice who receive a control antibody, they just have these large tumors that take up a large part of the chest cavity. And they're those kind of glowing green things. And GD2 antibody alone really doesn't do anything. You still have those large tumors and they're quite numerous throughout the lungs. CD47 actually has some effect in preventing the metastases. So you kind of see much smaller metastases and way fewer. But then when you use the combination of GD2 and CD47, you see this real clearance of all metastases from the mouse lungs. And this is just the quantification of the metastatic lesions that we found in the lungs in this model. And, there was a recent trial of anti-GD2 antibody in patients who had osteosarcoma that had undergone surgery to remove the relapsed lesions from their lungs, and then they tried to consolidate them to keep them in remission by giving anti-GD2. And it did not seem to work to prevent relapse, but we don't think that means that this approach of using anti-GD2 in osteosarcoma is going to always be ineffective, just that it seems that you need this boost on the macrophages in order to have this effect against metastatic disease. And then this I think was my last data slide, is just looking at the macrophages that are in these osteosarcomas after the treatment. So here you can see this is just staining for the number of macrophages. And you can see when you give a single antibody, you do increase the number of macrophages, it seems, compared to control. But when you give this combination, you just get this very dense macrophage infiltration. And then there are different types of macrophages. Some of them are kind of pro-tumor, anti-inflammatory, and some of them are anti-tumor. And so this INOS marker is a marker for kind of anti-tumor activity and this class of macrophages. And you can see that you really only see this flip in the type of macrophages you have when you give both GD2 and CD47. So, just to summarize this part of the talk is that we think tilting the balance between these eat me and don't eat me signals towards phagocytosis in pediatrics is really a promising approach because there are so many macrophages in pediatric diseases. And we really think we can take something that may be immunosuppressive and turn it to be anti-tumor. Anti-GD2 really synergizes well with anti-CD47, both because it drives upregulation of this eat me signal, calreticulin, on the cell surface, but also because it blocks this kind of new checkpoint, Siglec-7, that's expressed on immune cells. And we're translating this to a phase 1 clinical trial that we're hopeful will be open in sometime in April or soon afterwards. And that'll be open at 14 centers throughout the United States. Before I kind of do my acknowledgement slides, I also like to show a picture when I give these talks, especially to pediatric groups, of my friend Karen Glick, who died when we were in 7th grade. And the reason I show the picture is because when she died of CML, which was diagnosed in blast crisis. And at the time, that was an incurable disease. She died, actually a few months prior to the start of the beginning of clinical trials of Gleevec, which ended up being this miracle drug for CML. And now CML really is a curable disease because of these new technologies and these new drugs that were adopted. So when I think about my research, I really just think about, our goal is basically to, as quickly as possible, integrate these new technologies and drive these new technologies into trials for patients in order to turn diseases that were really previously thought of as incurable into curable entities. So just want to say thank you to all the people that did the work. Johanna Theraboth was really my partner in all of that GD2 CD47 work. My mentor, Crystal Makel, and a lot of that work was done while still I was in her lab. Of course, my new lab, this is the one outing we had prior to COVID, and then all the funding sources, and of course to Anne and Mib for inviting me today.
Yes, to your final slide with Gleevec, 100% on that. And before your final line in the slide before that, I had a question. If there are any GD2 and CD4 trials in neuroblastoma, or all of the GD4 plus CD47 trials in the lab only in mice?
Yeah, so that GD2 CD47, we are going to open that trial in both neuroblastoma and osteosarcoma, and that will be the first trial with that combination, and it'll actually be the first trial of any anti-CD47 in children. So we're excited both by the fact that we're testing a new agent in pediatrics, and we're being able to, I think we're getting smarter about the way we test these agents. We're doing, very short runs of the patient getting only anti-CD47 to look for safety for about 3 weeks, and then we'll very quickly bring in the anti-GD2. So we're excited that we're able to do a first in child that's also really a first in human of the combination.
Okay, great. Let's go. So you think like sort of April-ish? I know you're putting a date on it, but
yeah, I mean, that our goal is, we're supposed to be activated by mid-April. CTEP and the NCI keep a strict, kind of timeline, and of course, there's unanticipated delays, but that's really what we that's really what we're hoping for. And
okay,
if it's not, then it'll be soon after.
Please come back and tell us about it.
Absolutely.
Okay, questions.
When do you think is the right time to try a trial? If TKIs are keeping the disease under control, should patients continue taking them despite the side effects, or does it make more sense to consider a trial if one seems potentially positive?
Yeah, I think it's a great question. I think it's important to discuss with your doctor, and of course, the decision is going to be different for every family. And for every patient. Honestly, I actually don't take care of osteosarcoma patients that much in the clinic. I mostly take care of neuroblastoma patients, so I don't feel like I have enough expertise in TKIs. I mean, I know they have promise and have clear efficacy, and sometimes when you join a trial, it's really a promise of, a hope of efficacy from work that's been done in the laboratory. So I think this is very much something that you should, it's important to think about, but also to really discuss with your doctor and kind of figuring out what's best for a specific patient.
Is there anything anyone can do to help move this trial along so it can open sooner? Is there anything, funds, manpower, anything we can do to help support you and the funds to move things forward for these patients and families?
Sure. So I really appreciate that. I think there are definitely things that families and advocates can do in order to get more trials open sooner. In this particular situation, I think you're talking about the GD2 CD47. I'm not sure there's anything that's going to get it open quicker right now, just because of the way the machinations of NCI and where we are kind of with the timeline. And, we're only hopefully a couple months away, but I know a couple months is even very important when you're staring down a relapse. I think that brings up kind of larger questions about what's needed to push immunotherapy research forward for pediatrics specifically, what's needed to push osteosarcoma research forward. So, Anne told me I was going to get a question like this. Sorry, Anne, I had to blow your cover. But, so I jotted down a few things, just kind of started thinking, I think one thing is there's a lack of tissue from osteosarcoma and primary sarcoma patients. We get small biopsies done by the orthopedist, and we say it's because it's safer and easier to do with interventional radiology than kind of an open biopsy. But and I think this came up when we were at the conference, the Factor conference last year also. In the end, we need more tissue in order to, from an immunotherapy perspective, discover what's on the surface of these cells and what can be targeted. And so I think, we as investigators need to do a better job of figuring out how to get that tissue. In neuroblastoma, we probably could do a lot of diagnoses. Honestly, some of it now we do by you can figure out by circulating tumor DNA, and that's safer, but we're still doing open biopsies because we want the tissue, and that's the certified way to get the diagnosis. But I would say there's while there's maybe some risk involved in kind of open biopsies, I think the community and parental groups kind of have to look together to kind of create guidance around this. And I think it really would help to have more tissues available. So, that's kind of one thing. Obviously, I think, we were talking about also, what kind of funding or manpower could help. I think getting more people interested in pediatric research is really important. Funding through Factor and through MIB is wonderful because it's really targeted to osteosarcoma. And I think targeted osteosarcoma research actually could really help, especially with people that are already involved with osteosarcoma, saying, okay, this is something I want to support. All academics have to write grants and write papers, and it's part of what they do. And that's really never going to go away. But just taking some of that workload away, saying, okay, this postdoc is supported for the next 4 years in your laboratory, it takes away having to write even one less grant and think more about science. So I think something like that could be really helpful. And then I think we need to be strategic, thinking about targets that cross with adults in a way that makes sense for kids. Yeah. So, PD1, it would have been amazing. Everyone thought, okay, if it's going to work in one disease, it's going to work in osteo, but it didn't work. And also it wasn't tested in osteo until there was already an FDA approval of the agent. So I think we need to look, when we think about research done, we have to be realist. To develop an antibody drug by itself is very, very difficult, especially for a pediatric disease, right? And things are changing now because of really great advocacy and legislative work that's been done to change incentives. But in general, if there's targets that overlap with adult oncology, and you can find a researcher who's looking at those targets and get them interested in pediatrics, or even a target that is kind of like B7H3, that is just pan malignancy, both in pediatrics and adults, something like that, it's much easier to push when you think about a rare disease, which, frankly, is hard to get. To start an antibody drug, you might need $25 million for a clinical trial if you're making that antibody for the first time yourself. So I think kind of being strategic around that it could be really helpful. And then, obviously there's a lot of hope in for CAR T cells, and that becomes another big expensive endeavor for a different reason around cell processing. We're seeing the NCI start to respond. They have now, NIH will now be making CAR T cells for 2 different multi-site trials, one in AML and one in neuroblastoma and osteosarcoma. So that's great because they're doing something that probably, is much easier for the US government, is to make, a medicine out of nowhere, ship it, and indemnify themselves against any damage that it causes. But Seattle Children's has been doing some of this also. They've set up kind of a network where eventually they'd like to be shipping cells. And I think pushing more of those things, getting more manufacturing capacity and distributive capacity really could be transformational for pushing forward CAR T cell trials.
Yeah, agreed. A big part of the reason that we started Factor and bringing everybody under one roof is exactly what we're talking about, where physicians and researchers are, let's just take the bridge behind you, for example. Physicians and researchers are on one side and the patient and patient families are on the other side. And I think in both cases, we don't know what we don't know, to quote Confucius. And what I think physicians don't understand, in my experience, is that as a family with a child with osteosarcoma, if it were me, I would sleep under a bridge and pay whatever I needed to pay to get funding for whatever my child needed. And I am not alone in this. This is not this, this is not anything that's unique to me. The other thing that has come out in our Factor, in our outsmarting funding rather, is that many times the researchers and physicians think, oh, well, the patient's never going to go for that. But if the alternative is death, I mean, we're gonna do it. We're gonna do it. And I know there's a responsibility on the physician side and the researcher side, but the kids who have a seat at the table at our outsmarting meetings say, let's do it. Like, I'm here for it. And if I'm already suffering and I already know that the end is near for me because there are no more treatments for me, I will endure whatever I need to endure so that no other child should have to suffer as I do. Now, I know that there's responsibilities on the physician researcher side, as there absolutely should be, but there is that place to meet in the middle where we really can, and we really want to, and we really need to help the researchers, physicians, where it's imperative that we do. And it's the only way that we get out of and away from 40-plus-year-old treatments is by walking and meeting in the middle.
And I want to echo, what you say. Obviously, when we design trials, we can't do things that,
Yes.
would risk patients' lives.
Yes.
But we want to be bold, but be responsible. And I think that's a really important thing. And it's something that parents,
Yes.
actually, when we have parents in our corner, it's much easier to do that. I was at another meeting, which is kind of one of these collaborative 10-center meetings, and there was a parental advocate from one center and was like, why is there not a biopsy required on this trial? You learn so much from the biopsies. And we're like, well, we think, the IRB won't let it. And they're like, well, we do it at our institution. And the IRB, the parents were like, oh, we went to the IRB and said, that's ridiculous. You need to allow these biopsies.
Yeah.
And I think we need to change the culture around that.
For sure. Especially in osteosarcoma, we've already given body parts and said, yes, you can use it for research. Like, we got lots of stuff to give.
Yeah. I'm hopeful with CAR T cells. We'll see how things change over time. Right now, the virus that you make basically has to be clinical grade, the same virus that's made when it becomes an FDA approved product. And there is talk about at the FDA for phase 1 trials about kind of, obviously the thing still needs to be grade that can be used in human trials, but there's a kind of loosening that to make it easier so it can be made at an academic center and you don't need kind of this big, big pharma buy-in that may be a lot slower. So hopefully those changes happen, and we are, I think doctors working with advocates is one of the most important things about how we're going to keep moving the field forward and move it forward as quickly as possible.
For sure. And we know you're as anxious as we are to, we know that. We know you're as anxious as we are to get this solved.
Once the trial starts, how long does it typically stay in stage 1 if it starts in stage 1? And about how many patients do you treat who are in stage 1 in these clinical trials?
Sure. So it very much depends on the trial. Most of them are written as dose escalation trials. So a lot of the CAR T cell trials don't know what the safe dose is yet. So you start at somewhere that looks like it was probably safe for, let's say a CAR for leukemia, you do 3 patients. If it's safe, you may increase 1 or 2 times and do another 3 patients. And that usually is kind of the phase 1 dose finding portion. And you could take that and move to a phase 2, but sometimes when we design the trials, we do what's called expansion cohorts within the phase 1. And part of why we do that is it just lets us use the same trial and say, okay, we found the safe dose. Now we're going to treat 10 patients with osteosarcoma and 10 patients with neuroblastoma. And, there's several reasons to do it. One of them is it just removes the regulatory headache of opening another trial. The GD2-47 there, we know the safe dose of CD47. We know the safe dose, of GD2 in kids. We know the safe dose of CD47 in adults. And, we got permission from the NIH and NCI to use both of those doses as the first dose. We'll treat 3 to 6 patients, and if it's safe, basically then move right on to the expansion cohorts, in which case we treat 10 to 20 patients with kind of each disease type. If it's not safe, then we decrease the dose. So we're trying very hard, especially with agents that are known to be safe in adults to use extrapolate and assume we know what the dose is going to be safe and effective in humans, in children, and pull back from there after if we hit toxicity, rather than starting at these minuscule doses and moving up, because we don't think we should put any patients on a trial at a dose that we don't think would be effective.
And I have something from Oscar. He says, not a question, but just want to thank Charlotte and Mia for sharing your stories. Keep fighting. And thanks to Dr. Majzner for your amazing presentation today. Okay, with that, wrapping it up. In case you haven't been on social media today, you might know that it's World Cancer Day. And for this, MIV created a cancer card, otherwise known as the C card. We asked our junior board for their thoughts on this C card that was created. This was one of the responses, which I thought I'd share with you. Some people carry around a business card. We carry around our cancer card because it is our story. The membership is not revocable. It is not about freebies or good parking, because if any of us could trade the card in to never have had cancer, we would. It's about the courage, strength, and humility that having your C card shows. You can help us make it better. If you can make a donation in honor or in memory of someone who holds a C card, that would be great. It helps us do what we do, which we talked about at the top of the hour. With that, Dr. Brian Van Tine will be here next week of Washington University in St. Louis on osteosarcoma tumor metabolism. That's going to be a great one too. And thanks for joining us today. And of course, thanks to our guest, Dr. Majzner, for your work and your dedication towards better. Thanks to our panelists, Charlotte, Mia, and Amy. And thank you all for being here with us. We'll see you next week. Be sure and subscribe to our YouTube channel, where you can view our library of this and all OsteoBites topics and rockstar speakers. You can also listen to OsteoBites via podcast wherever you get your podcast.